John O'Keefe and Jonathan Jastrzewski found that there were these special neurons whose activity was strongly correlated with the animal's position in space. Let's do a virtual experiment. We will trace the animal's path as it runs freely around the cage, and at the same time stick an electrode in one of the hippocampal pyramidal neurons, recording its activity. Every time it activates, or in other words generates a spike, which is a fundamental unit of communication between two neurons, we're going to place a dot at the point in space where the rat was located at the time of the spike. You can see what happens. This neuron seems to be a huge fan of this particular patch on the ground. You can clearly see the neuron's preferred location. Due to such spatial specificity, neurons that exhibit this pattern of activity were given the term place cells. And corresponding patch on the ground where the place cell is active is called the place field. If you wirelessly recorded the hippocampal activity of a freely flying bat, you'd see that place fields roughly form spheres in three-dimensional space. Interestingly, even the change in non-spatial features can cause a portion of place cells to remap. For example, changing the odor in the room, or fear conditioning. If you apply mild electric shock while the mouse is running around in a familiar cage so that it develops the fear of this environment, that causes place cells to remap, even though nothing about the chamber itself has changed. So the internal map of a familiar restaurant you have been going in for years is drastically altered once you have a heart-shattering breakup at one of the tables. We can see how there is this additional layer of information about the context, not just the physical space. And remapping ties this information about physical location in space together with particular events and contexts meaningfully relating to that space. They can directly represent non-spatial information as well. Let's consider the following landmark experiment. Rats were trained in a sound manipulation task. They were first presented with the sound of a particular frequency. And to get the reward, the rat had to move the joystick which varied the frequency of a tone produced by a speaker. Basically, they had to make the frequency match the frequency of a sound they heard at the start. As you might have guessed by now, the investigators recorded the activity of pyramidal neurons in the hippocampus, the same type of cell that usually has a specificity to physical location. And here's the kicker. As the animal was varying the frequency, neurons seemed to have a preferred frequency, around which they had high pyrene rates, and showed almost no activity when the sound produced was in a drastically different range of frequencies. They behaved in exactly the same fashion as conventional place cells behave during foraging, having a well-defined place fields. It looks like hippocampus constructs and tracks continuous variables which are relevant to the task.